Alright, let's do a quick review. Cover all the bases of the different parts of the cell cycle. Look at those stages again of mitosis. Look at some pictures to be able to help us identify what we're looking at. Alright, this picture, we're looking at this picture. We know that about 90% of the life of a cell, okay, is going to be in interphase, where you have the G1, the S, and the G2 stages. The last little 10% is where it's going to start the cell division process, the nucleus division through mitosis, and then go through cytokinesis. Okay, so that's your interphase. Remember, G1 is where the normal life processes of a cell are happening. It's growing, it's carrying out processes like respiration and transport and everything else that it'll do. The S phase is where it's going to start replicating its DNA. That way, for the G2 phase, it'll prepare itself. Once replication is complete, it'll prepare itself for cell division. Then you have mitosis, and then the last is going to be cytokinesis, or the splitting of the cell. That's the cytoplasm. Alright, these are some good pictures. Okay, we saw pictures similar to this when we looked at our mitosis lab that we did. Okay, each cell we can see, and we can see the membrane around each cell. Here's just interphase. We can actually see the chromatin inside the nucleus condensing or showing up. Okay, and interphase is where the cell's carrying out its normal processes. We can see the nucleus and that type of thing. Prophase is where we see the chromatin starting to appear. You notice the spindle fibers here on each side have formed. Okay, that's a very good example of prophase. Metaphase, you see the spindle fibers on each end of the cell. Then you notice that all the chromosomes have lined up along the middle, and spindles have attached to those chromosomes. And then anaphase, this picture is more metaphase, because in anaphase what you're seeing is the separating. So I would call this one anaphase, not this early telophase. I would call this one anaphase. This one is still metaphase, so don't get confused with the wording or the labeling. Here would be your anaphase, where the chromosomes have been pulled apart and they are moving to the opposite ends of the cell. And then telophase, as you see, the nucleus is forming, and you can see the cleavage furrow forming here to split the cell through cytokinesis. Okay, I mentioned this, about 90% of the cell's lifetime is spent in interphase. And then during that interphase, it'll have those three major processes. It'll go through G1, it'll go through S, and it'll go through G2. Sometimes, however, there are issues that cells run into. Usually there are enzymes that kind of trigger, okay, it's time for you to start preparing your DNA for replication, and then, okay, it's time for you to start preparing yourself to divide because you're getting too big, and then, okay, it's time to start the nucleus division process through mitosis. There are times when this control isn't happening, and cells just continue to grow and proliferate and grow and proliferate, and there's no enzyme or protein to cut off that process. When this happens, this is usually a precursor to cancer. Okay, if you look at this process, you have these checkpoints, these proteins, if you will, that will check or regulate when it's time to stop one stage and begin the next stage, and when it's time to stop another stage and begin a third stage, and so forth. When those checkpoints aren't there, okay, that's when this problem comes into play. But we'll get back to that later. Let's go ahead and try to identify each stage. Okay, notice normal cell. You have a nucleus. That should be Interphase. Okay, when you see the chromosomes appear within the nucleus, the nuclear membrane starting to dissolve, and these centrioles starting to form, that is your prophase. When you see the chromosomes line up along the middle of the cell and the spindles are at the opposite ends of the cell, that is metaphase. When you see the chromosomes being pulled in opposite directions, remember we've replicated chromosomes, so now we have after DNA replication occurs, we have two sets of our DNA going on during these stages. And so when you see anaphase occurring, which is D, it's actually pulling a full set of DNA this direction and pulling a full set of DNA this direction in the cell. Okay, because you remember that DNA replication occurred before this cell division process started. That was in interphase. DNA replication occurs in interphase. So we have a full set of two full sets of DNA during prophase, metaphase, and anaphase. That way, when we get to E, notice you have two nuclei forming around those two different copies of DNA. The cleavage furrow, the pinching of the membrane, you have telophase and cytokinesis. Once you get your two final cells, they're going to contain that complete set of DNA, and it's an exact copy. Alright, so hopefully you can look at these pictures. We should be able to recognize when we see chromosomes and we see the the nuclear membrane dissolving and these spindles forming, that should be prophase. Okay. When you have your chromosomes lining up along the middle of the cell and spindles attaching to them, that should be metaphase. When you have your chromosomes being pulled to opposite ends of the cell by those spindles, then you're looking at anaphase. And then you have the pinching of the cytoplasm and the develop of your two new cells, your two daughter cells. Then you're looking at telophase and then eventually cytokinesis. This is a neat picture because what happened is in this captured by an electron microscope, they put these pieces together and they color coded them so you can see just what the nucleus or what the chromosomes in the nucleus look like through each stage. So you have just a regular nucleus and interphase. You notice that as you go, the chromatin starts condensing and forming chromosomes. All right, eventually it reaches a point to where those chromosomes have been developed in prophase. They start to line up along the middle in metaphase. They start to split apart in anaphase. Eventually they reach telophase where they have separated and the new nuclei are forming around them, and then you get your cytokinesis. So that's just a neat picture of looking at that path or that timeline. Okay, so let's just a few more review parts to this. There are terms we haven't looked at before, like diploid and haploid. Diploid means that the cell is containing a full set of chromosomes in its nucleus, where haploid means it's containing half of a set. Okay. And so this first question is the beginning parent cell, the original cell, is it diploid or haploid? Well, it's going to be diploid because it's containing a full set of the DNA. Question 2, the two daughter cells that form, okay, the two daughter cells that form, are they going to be diploid or haploid? Well, same thing. You should have complete sets of DNA in your nuclei. All right. How many daughter cells are produced? Well, that's easy because you're splitting one and making two. Are the resulting daughter cells different from the parent cell? No. Think about it again. If your skin cells die and you're making new skin cells, then you're wanting to make an exact copy. So you're wanting to replace the old skin cells with the new skin cells. So they should be exactly the same as the original cell. Are the resulting daughter cells different from each other? Once again, no, because if you're replacing old with new, you want it to be the exact same. And then how many chromosomes are in a human cell, an original human cell? Well, we haven't really got to chromosomes yet, but hopefully you remember that we have 23 pairs of chromosomes, okay, which make up 46 total. That's how many chromosomes we have. And then, but we'll get to chromosomes later, so don't worry just a whole lot about chromosomes right now. How many chromosomes are in each daughter cell? Well, remember, we made an exact copy before we went through cell division, so there's still 23 pairs or 46. What type of cells are produced by mitosis? Well, all of our cells are. Okay, whether it be heart cells or bone cells, muscle cells, skin cells, eye cells, everything that is structural is going to be produced by mitosis. Only the somatic cells, only those structural cells, not the gametes, not the egg and sperm, the reproductive cells, only the structural cells. And remember, a cell will go through mitosis for a number of reasons. For question 9, if it's gotten too large, okay, so it's using energy. If it's damaged and needs to be replaced, okay, if there's too much surface area for transport to occur, those types of things.